Hi, I'm Laney. I'm a biology major and I will be graduating in May of 2026.
So exciting. You only have 1 year left.
Yep.
What are you most excited about for your last year here at Capitol?
I think all of my TA opportunities. I'll be the head lab tech for the microbiology lab in the fall and the spring, and then I'll be TAing for microbiology and also research methods.
What excites you about being a TA? Is it working with some younger students? Is it advancing your career, or really all of the above?
I enjoy working with the students. I like seeing them kind of come in on day one and they're kind of out of place. And then, throughout the semester, you see a lot of them grow and some of them pick up micro and we try to tell them, like, maybe this is your calling. And also the work experience. I really love microbiology. I loved it when I took it as a student myself, and I've loved it ever since I've gotten the opportunity to TA as well.
Oh, were you one of those students who was kind of on the fence and then until you took the class, or
Yes,
did you know right away?
I was definitely on the fence. I had taken genetics the year before, and so I really liked genetics. My whole entire biology career, I've loved every class I've taken. Biology is one of those things. It's just my all-time favorite. But when I took microbiology, I think it also helped. The professor, Dr. Mohr, is like incredible. So he takes what we learn in lecture and it's very hands-on. So that was a great opportunity and it really piqued my interest.
So let's talk about your summer, because you had a very productive and exciting one.
Yes.
Where did you first hear about Summer Scholars and why were you interested?
So it was the spring semester of my junior year. Dr. Mohr and I were meeting for one of the Tri-Beta meetings, and he had proposed the idea of me applying for Summer Scholars. And I eventually ended up asking my mentor, Dr. Witzky, kind of asking her if she had any project ideas or if we could kind of bounce ideas off of each other. And we ended up landing on a project that looked into antibiotic resistance in E. coli.
Okay. And why was that exciting for you?
So I'm very interested in genetics, micro, cellular, molecular biology. So this project really encapsulates all of that, and it's more of a molecular genetics-based project.
What was the challenges, or were there any, kind of as you navigated what project you'd be on?
So originally the project that Dr. Mohr and I were looking into was actually a project possibly on primer, which he now is like the sole professor over. So we had originally thought about maybe doing that, but he had a couple other projects that he picked up, and so I didn't want to overwhelm him. So I ended up talking to Dr. Witzky, and I mentioned that he brought up Summer Scholars, and so then we had a meeting, and she was like, yeah, that'd be a great idea.
Good. Yeah. And then you had to write your proposal and submit it, correct?
Yes.
What was that process like?
A lot of Zoom meetings.
Okay.
A lot of articles to read. Because we're looking into E. coli and antibiotic resistance, the CDC does antibiotic resistant threat reports. So I've read through a lot of those, focusing on the antibiotics we used for the project and antibiotics that specifically target E. coli for our project.
Was that your first time really starting a literary review, and how did that look like, reading some of those CDC articles?
Yeah, so I've read a few articles for previous classes. We had some for genetics, we had some for research methods. It was definitely a different experience though, reading it on your own, because I think I had a lot more questions, because as a class we would go over everything. And so everything felt simplified. But when you're reading it on your own, it's a different story.
Yeah.
It's like you have to almost understand it yourself. So I would come to Dr. Witzky with quite a few questions about what were they talking about?
Yeah.
What does this mean?
But what were you excited about when reading some of those articles every week?
I really enjoyed learning about the importance of antibiotic resistance. So I've learned about it since my freshman year here at Capitol. We went over it in Bio 151, talked all about how you can get antibiotic resistance through eating meat if the animals are given antibiotics unnecessarily. We talked about it in genetics a lot. I think just having hands-on experience, really going through articles, for scientists that know this like front and back. That's all they study. So it was nice kind of learning about antibiotic resistance from people that's all their focus is. Yeah. And they just have so many like details and everything that just really put into perspective kind of the importance of our project as well.
Did it grow your passion for the field?
Yes. Yeah.
Okay.
Yeah.
How so?
Just getting to do this project hands-on. So, kind of when you're doing a science project, you have your hypothesis, you have your main question. For our project, it was, why is this gene important for antibiotic resistance in E. coli? And you're kind of hypothesizing, what do we expect to happen kind of thing. Mm-hmm. And seeing that happen was great. Seeing that hypothesis, supported was a great opportunity, and it really just amplified my love for research. So.
Can you tell us the title of it and what your hypothesis was?
Yeah, so we were looking into the importance of EFP in antibiotic resistance for E. coli. So EFP is a gene within E. coli's genome, and previous studies have showed its importance in antibiotic resistance. They know that it's needed for the resistance against certain antibiotics. So there's antibiotics called beta-lactams, and these beta-lactams target the cell wall of E. coli. Well, when that gene is there, they're able to produce proteins, and those proteins synthesize that cell wall. So it makes it harder for them to target that bacteria and kill it during treatment. So we ended up doing like a deep dive into a list of proteins from a previous study, which focused on 126 different proteins. And I got to do a deep dive into each protein and annotate it and figure out why it was important in the genome, because not all of them are the same mechanism. And I found 6 that were important for cell wall synthesis. Okay. So that was the focus for my project, specifically a protein called AMPG. And AMPG is needed for cell wall synthesis. EFP is there, it produces AMPG, and then AMPG is there for cell wall synthesis. So those beta-lactams have a harder time when EFP is present.
Okay. Is that what you thought was going to happen, or were you surprised along the way?
I had a couple surprises. So we were using 4 different antibiotics for this project. We had 2 carbapenems and 2 ESBLs, so extended spectrum beta-lactamase producing bacteria. Okay. Antibiotics. So our carbapenems were meropenem and imipenem, and then those ESBLs were amoxicillin and ampicillin. We were expecting similar results for our for our single control, for the wild type control. That's the E. coli that has EFP present in the genome, and then we had another control which was missing that EFP gene. So we knew that sensitivity would rise for the antibiotics, and then we had our 2 mutations. So we were expecting those through that wild type control and then those 2 mutations to be a lot less sensitive to antibiotic treatments. However, with 2 of our antibiotics that we used, we did not see those results. So I had to actually do further analysis and figure out why that may have happened. So that was kind of a curveball when we were looking at the numerical data. So I did a deep dive on those antibiotics and figured out that imipenem tends to work towards gram-positive bacteria, whereas meropenem is more effective against gram-negative bacteria. E. coli is gram-negative, so that might have been why those results worked better. Those numerical values came out to what they were.
You wrote your paper and then your presentation.
Yes.
What did that look like? Was this the first time you had presented the work? How did it feel to kind of be able to talk about all the excellent work you did?
Yeah, it was the first time that I had actually put a presentation together that wasn't for a class, so that was an interesting experience. We would do monthly meetups, all of the summer scholars and the mentors, and so there was, at one point we had a simple 5-slide presentation, just give an update on what you've been doing so far this summer, and mine was not simplified enough, and I got that feedback, and it was helpful though, because in the end, for this final presentation, I was extra careful that I explained things better, even if it wasn't maybe explicitly written on my slides. I made sure to go through everything that I did with this project as simplified as I could, to try and better explain it to an audience that might not know what I'm talking about. So that was a challenge, but it was also, I think, a good opportunity for me to kind of learn that not everybody that I'm going to present to is a scientist or knows what I'm talking about. So yeah.
Okay, what was the support you received from summer scholars?
A lot of support from Dr. Witzky, my mentor. Any questions I had, she was so quick to answer them. If we had to, if she wasn't maybe on campus, we would hop on a Zoom and try to figure out, how we could approach, these discrepancies. And Dr. Lam was also a huge support. He told every summer scholar along the way, if you need anything, let me know. So that was nice knowing that I had, that support behind me. And also the summer scholars were super supportive and provided really good feedback too.
Did you have all the equipment you needed here at Capitol?
Yes, the only thing that we had to take to OSU was for our sequencing. So they are able to do a technique called Sanger sequencing. We were able to see our sequencing data after we would do most of the procedure here.
You are looking at publications.
Yes.
What does that process look like, since this is the first time you're kind of going down that route?
Yeah, a lot of edits to my final paper. Very rough draft right now. It's complicating trying to put every little detail into the paper. We did so much this summer, and 10 weeks is a lot, but it's also very short once you're actually doing the project. So making sure that I put everything into this paper in enough detail. And also Dr. Witzky's been super helpful in that feedback that she's provided. And yeah, just making sure that everything makes sense along the way as well.
The 2025 Rust Belt RNA meeting.
Yes.
Tell me a little bit about that.
Yeah, so it's a conference held up in Cleveland. You stay overnight up there, and you'll put together a poster presentation, and a bunch of different people that actually know what you're talking about, they come through and they look at your poster and they talk to you and ask questions, and it's a good opportunity. There's different faculty members from different colleges that'll come by as well.
Okay.
So it's a good opportunity as a student, I would say, to be able to talk to different faculty from these universities.
Absolutely. And you're looking at grad school, correct?
Yeah.
Okay. Do you feel like this experience will feed very well into your application, but also what you're looking forward to for grad school?
Oh, absolutely, yes.
Okay.
OSU is definitely a high contender on my list for grad schools. I'm looking into their molecular genetics program, and research experience is very important for a lot of STEM-related fields that you might be looking into. But also, if grad school doesn't, happen right away, it's also just a good opportunity for careers outside of, my biology, my, bachelor's degree. So it's still a good opportunity to kind of understand and get to know better, like, what research entails. There's good and bad that happen, and it's more realistic when you get to do a project compared to doing it in a lab for class. Because in labs, I think for class, they tend to go very well. The results are exactly what you thought. Yeah, so at the start of my project this summer, I did a bioinformatics list. So it was that list of proteins from that previous study, all of which needed EFP for something, right? Some sort of mechanism. And so when I did that annotation, I had 6 proteins that ended up, being hits. Did AMP-G this summer, and then we intend to, this upcoming fall, go through the rest of the 5 proteins, and then follow a similar protocol to what we did this summer.
So if there are any kind of younger students who is looking into a research project, whether it's in the science field or really whatever major they're in across campus, and they may have heard of summer scholars, but they don't know too much about it, what would you tell them to either encourage them to participate, or maybe discourage, or what kind of should they be looking out for?
I would say it's important, firstly, the faculty member that you choose to work with. I think it's when you're in a class setting, your professor's there, right? You have all semester with them. It's great. When summer comes around, right, you're kind of on your own. So I would say pick a faculty member that's going to be as dedicated to the project as you are, is willing to help you along the way. That's encouraging for those ups and downs. When I faced some problems along the way, Dr. Witzky was not judgmental. She was there to help, let's think of a new way we can approach that. I would say the only maybe downside is lack of break, right? You don't really get that summer break that everybody else might be taking. But I think also it's a good opportunity to kind of expand your resume and, like I said, get that hands-on experience. It's very different going from a class to a big research project, or even just from class to lab. Yeah. This project is very much so important for antibiotic resistance and better understanding antibiotic resistance as it's a growing concern. And I would say that it's a really good opportunity, projects like this, to kind of deep dive into something that's being looked into but still nowhere near better, if you will. The project, the problem is not solved, right? So
Absolutely.
this was a good project for that.